Okay, so today we've got our 4 guest speakers, all either current or former graduate students and postdocs in social evolution. And we've got Chanchal speaking about sociality and caterpillars, followed by Ilias talking about competition in birds, Tova talking about collective decision-making, and Aaron talking about mating behavior in spiders. And so what we'll do is each of these speakers has a talk that's between about 10 and 13 minutes long. And I'd like to allow time, maybe like 2 minutes or so for a couple of questions between each one. And so I really encourage you, if you have questions, please either ask on the mic or in the chat. And I'll just keep an eye on time and kind of cut the questions off at some point and transition to the next speaker. Okay, so I can come out of my presentation. Oops. And we can turn it over to Chanchal.
Okay, great. I'm just about to share my screen. Okay, I hope it's visible to everyone. Okay, good morning, everyone. Thank you, Dr. Dakin, for having me here today for the presentation. In this presentation, I'm going to speak about some of my doctoral work that looked at the mechanisms of sociality in caterpillars. Now, social grouping is widespread in larval insects, especially larval Lepidoptera, also referred to as caterpillars. And while the benefits associated with group living have been widely studied, which include, for example, thermoregulation, predator defense, and feeding facilitation, the mechanisms underlying the formation as well as the maintenance of these groups are poorly understood. And this is what I investigated during my doctoral work, focusing mainly on 2 mechanisms, vibroacoustics and genomics. And I did that by using the masked birch caterpillar as my organism of work. It's a North American species that feeds primarily on birch and alder leaves. And here is a brief overview of the life cycle of the species. Here is a pair of adult moths mating, a cluster of eggs laid on a birch leaf, group of early instars, and a solitary late instar. Now, this is an excellent organism.
Sorry, Chanchal, I'm not sure if it's just me, but I'm only seeing your first slide still. It could just be something on my end.
Could everyone else confirm if it's the same problem?
Okay. It could be that you just have to go into the slideshow, or maybe I don't know if you have 2 monitors.
I think
we're seeing
screen.
Yeah, we're seeing PowerPoint.
Yeah, like your slide organizer part, not the full screen.
Do you see a full screen now?
We see like the PowerPoint program, so it's not in the slideshow mode.
I have it open in the slideshow mode. I'm not sure why it's not sharing in the slideshow mode.
Yeah, me too. What if you try clicking that slideshow button again, just see what happens. Okay, it must be like confused about which monitor is the main monitor. I don't know how to fix that. any
Me neither.
You have 2 monitors?
No, I just have 1.
Okay.
That's why I'm not sure what's happening here.
Is it Windows or Mac?
Mac.
Oh, okay. So maybe can you go into like system preferences and it's been a while since I did this.
I know I've been using Windows for my work, so I'm not sure what's happening.
One called displays.
Let me see.
But I'm not seeing it on mine actually.
Oh, I see
yeah,
it in my system preferences. And
so system preferences should be at like on the bottom, like the toolbar on the bottom, and one of the system preference options is displays.
Yeah, I found the display. It's just that I don't see any option to adjust it for
okay.
the presentation.
Okay, well, I'm not sure. One option that we could do is that you could just like navigate through your slide navigator, and we can see the slide. We can see the slides. We just can see like for some reason they weren't advancing because when you were playing, the show wasn't playing to us. But
What if I keep it open? Like, do you see a slide now? And
exactly.
If I move it, do you see a next slide now?
Yes, exactly.
Okay, it's probably just doesn't go into the presentation mode.
Yes.
Like it goes, but it just doesn't share in presentation mode.
Yeah, like
maybe I'll just maybe I'll just stick this away for now.
Okay, sure. Thank you.
Just so that it works. Sorry about this. I have no idea what the problem is. Okay, I'll maybe just start over. Okay, so I'm talking about some of my doctoral work that looked into the mechanisms associated with sociality in caterpillars. Now, social grouping is widespread in larval insects, particularly larval Lepidoptera, also called as caterpillars. Many benefits associated with living in groups have been previously reported. These include, for example, thermoregulation, predator defense, and feeding facilitation. However, the mechanisms underlying the formation of these groups or maintenance are poorly understood, and this is what I investigated during my doctoral work, focusing primarily on two mechanisms, vibroacoustics and sociogenomics. I did this by working on masked birch caterpillar. Now, this is a North American species that feeds primarily on birch and alder leaves, and here is a brief overview of the life cycle of this species. Here you see a pair of adult moths mating. Here's a cluster of eggs laid on birch leaves. Here is a group of early instars, and here's a solitary late instar. Now, this is an excellent organism to investigate the mechanisms of sociality in caterpillars, mainly for two reasons. One, these caterpillars have been previously reported to generate complex vibratory signals. And number two, we see both the group living early instars and the solitary behavioral states in these caterpillars, providing us with an excellent opportunity to conduct some comparative studies to identify the mechanisms underlying this transition. So using this, so there were two main objectives of my thesis. First, I tested the hypothesis that caterpillars use plant-borne vibration to form groups. And then I tested the hypothesis that differential gene expression mediates this transition from social to solitary in caterpillars. So for the first hypothesis, we first identified how the grouping changes across instars. And as you can see, instars 1, 2, and 3, represented by the blue, orange, and green bars, they began forming groups within the first couple of hours. Whereas instars 4 and 5, represented by the red and gray bars, they did not form groups throughout the 24-hour observation period. Interesting point here is that instar 3, represented by the green bars here, they are sort of a transition stage where the larvae begin dispersing from groups to form their own individual solitary shelters. Next, we looked into the different signal types that are generated by each instar, and we identified 4 different signal types. There is anal scrape, generated by scraping a modified seta-like structure called anal ore across the leaf surface. Then there is mandible drum, generated by drumming the mandibles on the leaf surface. Buzz scrape, generated by tremulations of the body. And then there is mandible scrape, which is generated by scraping the mandibles across the leaf surface. Now in instars 1, 2, and 3, all the 4 signal types were identified, whereas in instar 4 and 5, we did not see any buzz scrapes occurring. So after identifying these different signal types and the changes in grouping behavior, we looked into recruitment. But before that, I'm going to play a video where you will see these 4 signal types generated by the early instars. Okay, so now we knew there is a change in grouping behavior and signaling across the instars. Then we analyzed some laser Doppler vibrometer and video recordings during the recruitment events and made some interesting observations. Number one was that signaling is generated by the residents in 100% of the trials. And by resident, I refer to the caterpillar that finds a feeding site and begins constructing a shelter. Number two, the potential recruits also signaled. However, the signaling rate was much lower compared to the resident. Number three, the signaling rates of the resident, they seemed to increase as the potential recruit got closer to the resident. And number four, higher signaling rates by the resident resulted in faster joining by the potential recruit. An interesting point to highlight here is that anal scrape and buzz scrape were the two signal types that were generated at the highest rates by the resident. While anal scrape was generated at a higher rate when the potential recruit was farther, buzz scrape was generated at a higher rate when the potential recruit was nearer to the resident. Both of these signal types are strongly associated with feeding and shelter construction behaviors. So it's possible that these residents are using these signal types to advertise a shelter location or a feeding site to the potential recruits, leading to the recruitment event. So to summarize these findings, I'm going to play two videos. In the first one, you will see a solitary resident laying silk and constructing shelter and generating these anal scrapes. And now in the second video, you will see the recruitment event happening and the signaling occurring at the time. So overall, these findings suggest that vibratory signaling is used by these caterpillars as one of the mechanisms to form these social groups. Now, coming to the second hypothesis, we tested if differential gene expression is associated with the social to solitary transition. When it comes to social behavior in insects, there's a wide variety of studies that one may find. However, majority of these studies are associated with eusocial insects, like those of wasps and honeybees. But if you look at simple group versus non-grouping examples, we would mostly come across the locust examples. And these studies reveal that gene function is conserved across the species, and thereby providing us with a list of candidate social genes then that we can then test in a variety of organisms, including the non-model, like the one that I was working with. So using this knowledge, I conducted differential gene expression analysis using the RNA sequencing data, both from these groups, as well as the solitary behavioral states. And then this led me to the identification of over 3,000 genes that were differentially expressed between these groups and solitary states. And while majority of these were associated with developmental and metabolic processes, what captured our interest was the presence of these genes that have been previously shown to influence social behavior in other organisms. So I further investigated one of these genes, the octopamine receptor genes, to test its function in mediating this transition from social to solitary by using RNA interference method. Now, this gene was found to be upregulated in these groups compared to this solitary state based on the differential gene expression analysis. And by reducing its expression using the RNA interference method, we observed that it led to expedited transition from groups to solitary compared to the controls. So if you remember, previously I mentioned that third instar is when this dispersal happens, but in these trials, we used second instars, and by targeting this gene, we could actually expedite this transition. Overall, these findings suggest that there is likely a genetic switch associated with this transition from groups to solitary in caterpillars. So to conclude, these findings yield some interesting insights into the group living behavior of caterpillars, as well as the mechanisms associated with the same. And in terms of future work, we are at a very nascent stage of understanding this complex vibratory world of caterpillars, and there is a lot more that can be done to further our understanding of how these vibrations are used in a variety of functions, as well as how they are received by the conspecifics, because at the moment we have no idea how they are being received by the conspecifics. And I would like to thank everyone involved in this project. I wouldn't have been able to complete my doctoral work without the amazing support that I had from Dr. Jane Yak and all the funding agencies. With that, I'm happy to take any questions.
Okay, wonderful. Thank you so much, Chantal.
Thank you. Sorry about that technical glitch.
Oh, great. So Flor has a question.
Yeah, it's about the last part. Why did you decide to research this gene octopamine compared to the other genes that you mentioned?
That's a great question. To be honest, we could pick any of those, but octopamine has been shown to influence a lot of things in insects. For example, it's been shown to influence aggression. So it acts as a neuromodulator, a neurohormone. And we had some evidence from other organisms specifically looking at these receptors. So kind of based on that, we went with octopamine, but for sure we could go with other ones too.
Okay, and do you think it will have like the same effect as the octopamine? Like, do you think the other genes have an influence on the transition?
We would think so based on our differential gene expression analysis, because they were all found to be either upregulated or downregulated in the grouping stage compared to the solitary stage. But the problem is that most of the times a single behavior is regulated by a cascade of genes.
Yeah.
So while one could be easy to target, the others could have other impacts too. With octopamine, we did observe mortality rates and things like that. So we did come to a dose that was working for us. But for sure, in long term, it could be affecting other things too.
Okay, thank you.
Have time for another question if there is one. I have a couple of, if nobody has one right away. Can you like make them more social? So that's remarkable that you can alter the gene expression and change the behavior. Can you change it in the opposite direction?
Interesting question. So with RNA interference, we are trying to reduce the expression. We are not really increasing it. So we also call them gene knockout studies. So you may just completely remove the expression. But in the opposite direction, I guess what could work is CRISPR. But that's something we didn't explore due to the time limits with my doctoral work. But CRISPR is really popular these days, although I haven't come across any behavioral studies in insects. I have looked at studies targeting, for example, the butterfly wing pattern color. So those things. So not RNAi, but that's something we could go with CRISPR likely. It would be cool to see if we could switch the solitary to groups.
Okay, one more I have, unless there's a student question. Do the recruits help with the shelter construction?
Yes, so that's another project that actually I think someone in Dr. Yak's lab is doing on how the groups are when they're formed. So based on my observations and the recordings that we did, they actually participate in all these behaviors. That's where the cooperation comes in. So for example, one could be feeding, other one is constructing. So they kind of take turns when it comes to the responsibilities of being in the group.
That's really neat.
It is, it's very interesting.
Yeah, I hope I can go out and look for them, look for caterpillars doing this myself this summer.
Yes, absolutely.
Any other questions before we go to Ilias? Okay, so I think we also solved the problem for Tova and Aaron when you're up. I think the issue was that make sure when you share your screen, you share your whole monitor, not just PowerPoint.
Oh, I see.
You were sharing PowerPoint and then the presentation was playing, but not to us.
Yeah, and clearly my animations didn't work because I was not in the presentation mode.
Exactly.
But I hope everyone understood what I was talking about.
Yeah, this is a good lesson for the students because they have to do presentations next week too.
Okay, great. Thank you.
Thank you so much, Chanchal.
Thank you. Okay.
Okay, so let's see if this works. Share screen. Share. Hopefully that's good. 1, 2. Okay, sweet. All right. Well, good morning, everyone, and really excited today to be here with you guys and present some of my research on animal sociality and competitive interactions. So with regards to animal sociality, I want to touch up on some of the benefits that it may provide species in both ecological and evolutionary perspectives. For example, sociality may improve foraging success of the species. It may increase their mating frequency and also provide opportunities to reduce the rate of predation. However, there may be possible costs of sociality for these species. For example, sociality may increase the rate of within-group conflict. So with the ideas of benefits and costs of sociality, I was interested to look at both of these sides of sociality through the lens of competition, animal competition, specifically looking at resource competition at backyard bird feeders, which I'll get to momentarily. My main question is to quantify the consequences of sociality at both the among and within species levels of competition. So while there has been some research in the context of within species competition, there's some research on the consequences of sociality, there's not as much insight into the among species level. So there's this knowledge gap that I'm hoping to address with this research here at this level of competition. So with this big question, I'm breaking it down into 3 components for the sake of this presentation. For the first component, I aim to describe species sociality. Here I ask, do species differ in their level of sociality? For the second component, I'm interested in looking at the consequences of sociality at the among species level of competition. So for this component, I separated it into 2 halves. For the first half, I'm asking, is there an effect of the presence of conspecifics on the outcome of interspecific dominance interactions? And then for the second half, if there is this effect related to a species' degree of sociality? For the third component, I then go into the within species level of competition and seeing the effects of sociality at this level. Here I ask, how often do conspecifics compete with themselves in a social context? So before I present the results of these questions, I wanted to take some time and talk about the data itself, because I think it's an incredible data set that I had the opportunity to work with. So the data set is called Project Feeder Watch. This is a program that consists of volunteers monitoring bird populations and recording bird observations during winter feeding times across North America. So here volunteers partake in 2-day recording periods where they record a lot of insightful variables, such as information on the time and then the location of their sampling period, information on maximum number of individuals observed per species, at one given time during this sampling, which I'll talk about a bit more later on, and also any occurrence of what are called displacement events. So these are events where there's a resident individual at a feeding location just foraging away, but they are caused to displace or to leave this location due to the presence or the incoming competition of another individual. So to hopefully better visualize this, I have this example here. So this is a recording that was recorded through this Cornell Lab observation program, interaction between a hairy woodpecker versus a blue jay. So here we see the blue jay is foraging away at this location site, but there's this hairy woodpecker that arrives. And this woodpecker wants this resource to themselves. There's a bit of a back and forth between the two. And ultimately, we see the blue jay is displaced by this hairy woodpecker. And now the resource is all for the woodpecker. So we see that in this case, the winner is the woodpecker and the blue jay is displaced. So this is an example of one of these displacement events. Sometimes they're not as intense as this one. So in this data set, we have a total of 196 bird species with over 9 million observations and around 89,000 displacement events. So just as I mentioned, this is a very rich database that can provide us a lot of insight to the questions that I'm hoping to address. Okay, so for the first question, I wanted to quantify species sociality and any variation that may persist in this trait. Before I did this, I subsetted this huge database into instances or species that have at least 100 displacement interactions. And this yielded 68 focal species for the upcoming analyses. So for each one of these focal species, we quantified their sociality, using this measurement of group size. And as mentioned before, group size is defined as the maximum number of individuals for a given species observed at any one given time throughout a sampling period. So here, I'm just going to move this a little bit here. Here we have a figure that sort of presents just how much variation there is in this measure of group size. On the x-axis, we have the date of the sampling period. On the y, we have the average group size. And each one of these lines represents a particular species in the data set. So here we just have a subsample of 7 species, but we can visually see just how much variation there is in group size across these seven species. So this is interesting for us because it allows us to ask questions on how and why such variation has persisted so far in evolutionary time. So with this data, we calculated group size repeatability for these species using a bootstrapping approach. And we were able to find that 36% of the variation in this group size trait can be attributed to differences among species. Furthermore, we were interested to see how, if there's a phylogenetic signal or an effect of relatedness across species with regards to the group size. So what we did is we calculated each species' average group size. And here I'm presenting these group sizes on a phylogenetic tree. So we can see here that there is an effect of relatedness. Species that are closely related tend to share similar group sizes and differ significantly, differ a lot from other species that are a bit more distantly related. So for example, here we see the woodpeckers and the hummingbird species tend to have smaller group sizes, whereas sparrow species down here in the bottom right and finches in the bottom left tend to have more medium to larger group sizes on average. So this just shows again just how variable this average group size trait is in our database. Okay, so we've identified and measured the variation and the group size averages for these species. Now we want to ask, how does sociality influence competition? So for the second part, we're looking at the among species level. But before we actually do this comparison, we need to first quantify dominance traits. And we quantified 2 traits, which I'll get to shortly. But the way that we did this is we used these species-specific models. So each species had a specific model, which consisted of a competition outcome variable as a response variable. This is a binary variable of either win or loss, as a function of 2 fixed effects, group size and relative body mass, as well as a random effect of location. So using these species-specific models, we quantified 2 dominance traits. The first one is competitive dominance, which is defined as the probability of displacing a similarly sized heterospecific competitor. The second variable that we quantified was the conspecific effect. This variable is defined as the average effect of nearby conspecifics on the outcome of a heterospecific competition. Okay, so with regard to the first trait, competitive dominance, we see this really strong negative effect. So I'll take some time to go through this figure here. On the x-axis, we have relative competitor mass. On the y-axis, the probability of winning an interspecific or a heterospecific competition. Each line in this figure represents each of our 68 focal species, which is colored based on their average group size. So we see here in general, the general trend is that as your competitor increases in body mass relative to yourself, you have a lower probability of winning a heterospecific interaction. However, with regard to the effect of conspecifics, there's a bit more ambiguity here, or a bit more variation. So here we have a similar graph, but instead of competitor mass on the x-axis, we have group size. So we see here that as group size increases, some species gain an advantage or have an increased probability of winning. Other species gain a disadvantage or a decreased probability of winning. But on average, across all species, there tends to not be as strong of an effect of the presence of conspecifics on competitive outcome. So now that we've quantified these two dominance traits, we want to see how they're related to species sociality. So again, we ran two regression models here, one for each of the respective dominance traits, both of which had fixed effects of group size and body mass. And what we found is actually pretty interesting. With regards to competitive dominance, we see here in this first figure with the x-axis of average group size, the y-axis of probability of winning, we see that more social species tend to be less dominant in heterospecific competition. Additionally, we see that in this figure to the right, larger species tend to be more dominant than smaller species. However, when it comes to the conspecific effect, we see that species that are more social have this positive effect of conspecifics. So this means if they're in a group with other conspecifics, they gain a competitive advantage, which highlights this potential benefit of sociality in the context of among species competition, which is really interesting. However, we need to look at the other level now. Now this is at the within species level. Here we were interested in figuring out whether there is more than, equal to, or less than levels of conspecific conflict compared to what we'd expect. And so for the sake of time, I won't get into the generation of this null model that we used, but we used a null model to generate our expected rate of intraspecific interactions, which is demonstrated here on the x-axis. And we compared this to the observed rate of intraspecific interactions. Sorry, these aren't rates, these are actual counts. But we found that with this comparison, 62 out of the 68 focal species interacted with conspecifics more often than expected. Furthermore, we found that the greater proportion of intraspecific interactions occurred among the more social species. So this shows us that even though there's this benefit at the among species level of sociality, at the within species level, social species tend to have this higher level of conflict between one another as opposed to less social species. So some final take-home messages. We were able to find strong evidence that suggests sociality differs across species. We found that while there is variation in the effect of conspecifics across species, with the effect of conspecifics on the outcome of heterospecific competition across species, there is this trend that more social species gain an advantage when it comes to the presence of other conspecifics with regards to their competitive outcome against heterospecifics. And also at the within species level, we see that there's more conspecific competition than expected. And this may be driven by potentially high competition levels during winter foraging due to the scarcity of these resources at that time. So thank you for watching this. I hope you learned something and are excited about this research as much as I am. And I'm open to take any questions that you may have.
Okay, thanks so much, Elias. Time for one or two questions as we transition to Tova. Great presentation. Sorry, thank you, Anna. I guess I can ask one if there aren't any student questions or other presenters that have questions. That interaction you showed between the woodpecker and blue jay was especially drawn out. That was a really good one. Do you think most of the displacements are like that? And what do you think it means if an interaction is prolonged like that?
Yeah. So from the videos that I've seen and the information that I've read, it seems like these displacement events are very quick. So it's individual is foraging, and then as soon as the presence of another individual comes, if that new individual has the ability to displace the resident individual, it happens right away. If, just like we saw today, if there's a more prolonged interaction, it might be that the two individuals that are partaking may be similarly competitive enough, so that there's a less difference in their competitive ability. So they think that, okay, it's worth me to fight a bit more because this opponent that I have may not be as strong as I think, or maybe similar to me. So I have a greater opportunity to invest, a great opportunity to win this resource. So I'll take the time to invest here. Whereas, like I mentioned before, these more instantaneous ones may be just a result of, okay, there's a way bigger individual coming in, or way stronger one, I'm going to get out of the way. Or I'm a bigger one, I'm going to come in and take this resource, and they're not going to fight me.
Yeah, okay, great. Thank you. All right, so just there's a question from Anna. Okay, is there a learning curve at all?
Oh, interesting. So like yes, so that, okay, so specifically for age, there's research that shows that experience and age in turn adds this increase in dominance in the literature. So as you age, as you gain more experience, you tend to be more dominant, more successful in these competitions than you were at the younger or less experienced time. So this occurs at the individual level. Now, there's also variation in this too, right? Because in some individuals within a species may be more dominant than other individuals. So this variation may be very, I think it's interesting to monitor an individual throughout their life to see how their dominance ability or the competitive ability changes throughout this age or experience time. So yeah, there's definitely a potential for a learning curve here. Whether this is similar across all individuals of the species, I'm not sure. And I think that's a really cool question to ask.
Okay, thank you. All right, so let's pass over to our third presenter, Tova, talking about collective decision-making.
Okay. So if I share my screen, can you see this?
Yeah, we have just seen a presentation, yeah.
Okay, and if next slide, you see that? Lovely, okay, perfect. So, let's see. Okay, so hi everyone, my name is Tova. I'm a PhD student at U Ottawa, but today I'll be talking about my Master's of Science research, which I did at McMaster University, where I was studying the effective experience on collective decision-making. So I'll start with a brief overview of collective behavior. So collective behavior is when a group of individuals has to coordinate their behavior to achieve a goal. And we see collective behavior in both humans and non-human animals. So for example, a flock of birds traveling together is collectively behaving, or an assembly team that's building an airplane. And common goals that groups are trying to achieve by coordinating their behavior include navigating and traveling throughout a landscape, finding food patches and foraging, staying vigilant and avoiding predators, and just overall problem solving. But to collectively behave, the group needs to communicate. So individuals need to share information within the group. And groups must also make collective decisions. So social groups are faced constantly with situations where they're required to make a decision. Should the group leave or should they stay? Which direction should they travel? Where should they build their nest? Should they fight the predator or should they flee? So in nature, groups are faced repeatedly with similar decisions, but we know little about how decision-making changes over time. So I wanted to know how experience affects collective decision-making. So to study collective decision-making, I use this well-established decision-making behavior called emigration, and we see this a lot in ants. So emigration is when a colony of ants searches and moves into a new nest if theirs becomes inadequate. So if theirs is damaged, for example, like in this photo. So we're using specifically acorn ants that tend to live in acorns. And in nature, decision-making is a large part of emigration because there's many possible nest alternatives around, but only a few nests would be high quality and durable enough that's suitable for their colony to live in. And colonies must also choose a nest quickly because they physically carry their queens and all of their young and their brood into that new nest. And that makes the lifeline of their colony extremely vulnerable to predators and environmental conditions when they're actually emigrating. So I'll briefly overview this emigration process. It's divided into 3 phases. And in the lab, the ants reside in these artificial nests made out of balsa wood and glass slides. And emigration begins when we remove the roof of the nest, making it uninhabitable. And the first phase is the discovery phase, where individual scouts will search for new nest options. And then once they find a new nest, the assessment phase begins. So this is where scouts will assess the nest based on characteristics like cavity size, cavity height, entrance size, and darkness. If the nest is good quality, the scouts will recruit other workers to also assess that new nest, and then they will accept it. And then this third phase is the transport phase. So once enough scouts have accepted this nest, the transport phase begins, where the scouts will physically carry the young, which is the brood, the queens, and all of the remaining colony members into that new nest. So only a minority of the colony members actively partake in the emigration. Most of them will be carried to that new nest. And we can make this emigration task more difficult by providing colonies with 2 different nests of various qualities. So surprisingly, little research has explored how experience actually changes this emigration process. So we know that repeated experience emigrating can increase the overall speed of emigration, but this has only been demonstrated with a single nest option available. So in our study, we tested if repeated experience emigrating and collectively deciding between 2 nest options can improve the group's decision-making ability. So our hypothesis is that experience with collectively deciding between 2 nest options will improve group performance on future decision-making tasks. So to test this, we first run a training phase where we provide colonies with experience emigrating with either a binary choice or a singular choice. So we have 20 colonies in the choice group, and they always receive 2 nest options of different qualities. So there's a good nest and a mediocre nest. The quality of nest varies on the amount of light that enters the nest cavity. So these acorn ants prefer darker nests because a darker nest is more similar to an acorn. And then they also reside in the standard nest that has no light filter on top. So it's kind of like a poor quality nest. And then we have the no choice group. So the no choice group gets a single nest, either a good or a mediocre quality nest. So there's no room for decision making here. And both groups undergo 6 emigrations during training. And we randomized and counterbalanced the order and location of the nests in the arena for each emigration. And every trial is spaced apart by 2 days. And then we test both groups on the same decision-making task, which is the choice between a good and a mediocre quality nest. So for the choice group, this is their 7th emigration with a choice. Whereas for the no choice group, this is only their 1st emigration with a choice. So in total, we have 2 measures and 4 predictions. So our first 2 predictions are about efficiency. So we predict that the choice treatment will be more efficient than the no choice treatment during the final test emigration. We also predict that throughout the training phase, the choice treatment will improve their efficiency as they gain experience. We measure transport efficiency, which is the proportion of transports directly from the standard nest to the good quality nest. So this captures transport efficiency. This is the most efficient way they can transport to the better quality nest, as opposed to transporting directly to the mediocre nest or from the mediocre nest to the better quality nest. So because we need two different nests to measure transport efficiency, we can measure it for both the choice and no choice groups during the final test emigration when they both have two nest options. But throughout the training phase, we can only measure it for the choice treatment because the no choice treatment only has a single nest option. And the other two predictions are about speed. So we predict that the choice treatment will be faster at decision-making than the no choice treatment during the final test emigration. We also predict that both treatments will become faster at emigrating throughout the training phase. And to measure decision-making speed, we're using something called decision latency. So this is the time between the first discovery of a new nest, including the assessment of that nest, and then the acceptance of that nest, and then the first transport of a colony member into that new nest. So I have some results to share. So in this figure, we have transport efficiency measured as a proportion on the y-axis. And then on the x, we have treatment. So we have choice in orange and no choice in green. And we found that the choice group had significantly higher transport efficiency than the no choice group during the final test. So this supports our prediction that the choice group would be more efficient during the test than the no choice group. In this figure for decision latency, we have decision latency measured in minutes on the y-axis and colony treatment on the x. And we failed to detect a significant difference in decision latency between the colonies. So we had predicted that the choice group would have a shorter decision latency than the no choice group, but we failed to detect that. So this means that both treatments, when given two nest options, experience collectively deciding between two nests did not increase the decision-making speed compared to colonies with only experience choosing with one nest. So now we're looking at transport efficiency throughout the training phase. In this figure, we have transport efficiency again as a proportion on the y, and then emigration on the x. So we have the 6 emigration trials. And we failed to detect a significant change in transport efficiency over the course of the trials. So this means that we failed to find support for our hypothesis that the choice group would improve their efficiency throughout the training phase. But we do see a lot of variation within each emigration. So one explanation is that there's no disparity in feedback that the ants receive during the training trials. So feedback is an important aspect of learning. Human social groups tend to learn more quickly when they receive feedback for their accomplishments and their errors. But for these colonies, the trials were spaced apart by 2 days. So regardless if they chose the good quality nest or the mediocre quality nest, their nest got destroyed after 2 days. So maybe if we varied the feedback between the qualities of nests, there might have been a clearer learning curve. So for example, maybe if the colonies received an extra day of rest, if they chose the better quality nest, they would have learned to choose the better quality nest more often. And lastly, we predicted that the decision latency would decrease within both treatments throughout the training. So we ran 2 separate statistical models, one for each treatment, and we found that the decision latency did significantly decrease over the course of the trials for both of the treatments. And we had expected that the choice treatment would have a longer decision latency compared to the no choice treatment because the choice treatment has a second nest throughout the training phase. And we expected the nest would take more time to assess and thus more time to make a decision. So that's why we ran 2 separate models. But surprisingly, when we looked at the figures, we decided to run a test post hoc to test if there's actually a difference between the groups, because there appears to be no difference in decision latency throughout the training for between the groups. And we failed to detect a significant difference of decision latency between the treatments. So basically, it takes the same amount of time to make a decision when there's one nest option there compared to 2. So because decision latency is not different between the treatments, it could be that the colonies are favoring speed over efficiency. So both the good and mediocre quality nests are acceptable to the ants. They're above their acceptance threshold. So both the nests are darker than the standard nest, which is better quality. So the colonies are upgrading when they emigrate, regardless if they choose the lesser quality mediocre nest. So basically, these colonies could be favoring the speed of choosing a nest, not whether they're choosing the better nest of two good options. So to summarize and conclude, our studies suggest that social groups can improve their decision-making ability as they gain experience. We also found evidence for the speed accuracy trade-off during emigration, as the colonies were favoring the speed of emigration because both of the nests were acceptable qualities. And this is evolutionarily relevant because social groups are repeatedly faced with similar decisions. And our research leaves us with more questions than answers. And a major question that we have is, how is collective decision-making tied to fitness? So we clearly predict that better decisions lead to higher collective fitness, but this requires future research to confirm. So I'd quickly like to thank the people that have helped me out on this project. And thank you all for listening. I will take any questions.
Oh, that was great. Thanks, Tova. It's so nice to be able to experimentally manipulate these behaviors. Okay, so we have a question from Chloe. Can you see it? Or I'll read it out loud for the recording, actually. Do we know why most of the colony is carried when emigrating to a new nest?
Great question. So ants have a division of labor. So there's some ants that are workers where they're like finding food, they're finding new nests, they're taking care of the colony. And then we have other ants that are nurses, and they're taking care of the brood. So the brood is the young, the eggs. The most important part of the colony is the brood and the queen. So the reason that most of the ants are actually carried there is because it's not their job to move the colony. Their job is to take care of the brood. So they actually, when they're emigrating, the brood always have to have like a babysitter with them. So first they transfer like some of the nurses, and then they'll transfer the brood, and then they'll transfer the rest of the nurses. So yes, it's like, imagine it like it's a baby. There always has to be like an adult around to take care of them.
Okay, that's so cool. Thank you. All right. Any other questions from students? I have one more that I want to ask if no one else has one. Oh, have you considered whether, and I don't know if it makes sense because of the like kind of relationships, the familial relationships that might happen between the colony, but if you change the composition, like kind of the level of experience with your teammates, could that be part of what determines some of these metrics of performance? Yeah, that's a great question. So it's actually very interesting because when talking about group expertise, which I kind of left out of this presentation because of time, but when a group improves, the knowledge and that improvement has to be stored somewhere. It's either stored in like the brains of individuals, or in humans, it's stored in like, you know, technology or checklists or equipment. So it would be really interesting to kind of split up a colony and see how many experienced individuals can actually like help the whole group achieve better performance. And that would be, so I was definitely considering testing that question if I stayed and did my PhD at McMaster, but I've switched to bees now. But yeah, that's a great question. Okay, very nice. Okay, one more question, and then we'll go to our last speaker. So Donovan has a question.
Hi, thank you so much. That was very interesting. I was just wondering if the response time, I guess, to when an ant's nest was destroyed was observed to change during the experiment. So
I'm the one who destroyed each of the nests. So it was like experimentally, I would remove the roof of the lab nest, and then like that's when we started the trial. So like before the lab nest was removed, we didn't count that as part of the emigration.
Okay, very, very interesting. Thank you.
Mhm.
Okay, great. So we're going to turn over now to our final speaker of 4, Erin, who's going to tell us about mating behavior in spiders.
Okay, so before I start, can everyone see this screen? Does it look like the full screen slide?
Yes, to me.
Excellent. Okay, and it switch to the next one. Okay, cool. All right, I'm just gonna set a timer for myself because I tend to go long. All right, so today I'm gonna be telling you guys a little bit about some animals that actually aren't terribly social, to the point where they actually will cannibalize each other. But I'm going to tell you about the social behavior that they do have, which is their mating interactions, and how that can have some really important impacts on their evolutionary trajectories, and also how abiotic factors can influence those behaviors. So we're kind of looking at proximal mechanisms of how we can see downstream effects. So just to kind of introduce this, we all know that animals have to deal with temperature. We are animals ourselves, and we have a bit of an intuitive sense for this. And we know that extremes in either direction of temperature can pose challenges. But temperature also has another slightly more subtle effect that we don't experience so much as endotherms, and that is that temperature directly alters the rate of chemical reactions. And since you can think of metabolism as a giant series of chemical reactions, temperature directly affects the metabolic rate. So we can control our metabolic rate because we can control our temperature. So we don't deal with this so much. But animals that are ectotherms and can't control their metabolic rate are kind of at the mercy of ambient temperature. So if we want to study temperature and animals that deal with temperature, maybe we want to go someplace where we experience extremes. And you might say, oh, the desert is a great place because the desert is hot. We have extreme temperatures in the desert. And in the place where I did my PhD work in southern Arizona, you can see that indeed it is hot there. So you'll often see scenes that look something like this, or biologists that look something like this. It is very, very hot. However, if you go at a slightly different time and in kind of an unusual year, you can also see scenes that look something like this, which is more of what you might see in Ottawa or me in London, lots of white stuff on the ground. So really, when we say the desert is hot, that's a bit of an oversimplification. What the desert really is variable. So animals aren't only experiencing extremes in temperature, they're also experiencing really large swings. And we can see this by looking at a plot of temperature in one of my field sites over the course of the month of April. The solid black line represents mean daily temperature, and the pink areas are minimum and maximum. So there's these giant swings in temperature every single day, and the animals that live there have to deal with this. So what animals do we want to study? There's lots of cool reptiles, my herpetology colleagues tell me. There's lots of cool birds, but I'm going to make the argument that jumping spiders is a really good study system for a lot of reasons. I'll give you a brief introduction to the reasons why I think they're great. So there's 110 species, that number is increasing almost yearly. It's one genus, and there's a ton of different diversity. They're hyperdiverse and recently diverged, which has some interesting evolutionary implications. They can be either habitat generalists or specialists, so you can set up some really interesting contrasts between species. And they're small. So I say this for 2 reasons. One is that you see these beautiful zoomed in pictures of spider faces and you think they're like the size of a golden retriever, but actually they're something like that. So not only are you now appropriately oriented to the size of these animals, but you can imagine that if you put this animal at a different temperature, it's going to equilibrate very rapidly. So temperature really is going to have an outsized effect on these animals. And the species that I'm going to be talking about primarily today is this one right here, Habernatus clypeus. They're in this deserty area, and also we had a whole bunch of them in the lab when I started my PhD. So those are the guys we're going to be talking about today. So my PhD worked something like this. I was interested in how desert animals interact with temperature. I looked at this a variety of different scales, from physiology all the way up to what I call landscape level processes. The bulk of my talk today, I'm going to be focusing a little bit on physiology and also interest-specific interactions, so those behavioral interactions between members of the same species. So just a very brief view into the physiological studies that I did. I was interested in how the animal's physiology was affected by temperature. And if you haven't had a physiology course before, this curve is very important when you're looking at animal physiology. It's known as the thermal performance curve. So you have temperature on the x-axis, and then on the y-axis is performance. Performance is broadly defined. It could literally be anything an animal does. And you can ask different questions about how an animal functions at different temperatures by looking at different parts of the graph. I'm not going to go into a lot of detail because I only want to focus on one question today, and that is, what temperature do the animals prefer to spend their time at? So there's some kind of range known as their thermal preference range where animals want to be. And that's what I'm going to evaluate for the sake of today. So in order to do this, we set up a thermal gradient. So we have this rig where there's heating elements on one end, and then the other end is cold by putting it in a refrigerator room that we had nearby. And it sets up this really nice gradient. So it went from 60 degrees all the way down to 15, and we released spiders into this gradient and then evaluated them and asked the spiders, what temperature would you like to be at? And we found some really cool things. First of all, spiders have really strong thermal preferences. So given the opportunity, they want to be right around 35 to 40 degrees. And that was 50% of all individuals. So they really have strong thermal preferences. But there's something even more interesting that happened. And that is that males and females have slightly different temperature preferences. Males prefer to be at ever so slightly higher temperature than females. And I want you to just kind of squirrel this away in your memory bank because this is going to be important a little bit later. So now I'm going to jump into the intraspecific interactions. And by this, I mean sexual behavior, because that is primarily the social behavior that these guys have. So long story short, I'm just going to let the spider show you what they do for their intraspecific interactions. The male spider is on the right here, and the female is on the left, and the male is producing courtship signals. So he vibrates, much like the caterpillars, which is very cool. They vibrate their body parts together to produce a vibratory signal that travels down through their legs, through the ground, and up into the female's legs. She has ears with her legs. You can measure the sounds using that laser dot on the abdomen. And then he does visual things too. And the female then decides if she wants to mate with him. So just quick overview of how we did this. We took courtship arenas and we warmed them up on the very sophisticated electric pancake griddle. When they were warm, we put spiders on there, monitored their courtship interactions. And then for the cold treatment, we took another courtship arena, put it in the sophisticated lab freezer. And then when it cooled down, we performed courtship trials there as well. And then a couple of different things could happen. The female could either decide that she wanted to mate with the female. The only kind of cue that she gives us that she would like to mate with the male is mating with the male. And they only mate one time. So we had to do a lot of different trials because each pair could only be used one time. And then if she doesn't like the male, she would just reject the male, either run away or occasionally attack. Here are the results that we have. So I'm showing a plot of across temperature, one specific aspect of the male song. So the buzz frequency, the pitch of the buzz. And I'm going to show you what it looks like at different temperatures. So this is what this is showing at cold, room temperature, and warm. And I'm also going to play sounds for one individual at 3 different temperatures. So in the cold temperature, it sounds like this. In room temperature, it sounds like that. And then warm it sounds like that. So you don't even need to do statistics on this stuff. It's very clear. And an interesting thing is if we superimpose what we know about their thermal preferences, it sits right there. So the males are singing at high pitches very vigorously where they want to be, which kind of makes sense. Males are nice and vigorous, exactly at their preferred temperature. So if we then look at the mating trials where we evaluated what females liked, we can see that females had higher mating rates in the warmer temperature. So the females are potentially being less choosy at this higher temperature. And I just want to show you this again, which is our temperature preference graph. So males want to be not only where their songs are high frequency, very vigorous, but also where females are less choosy and more likely to mate with them. Whereas females prefer to be at a slightly cooler temperature. So we see the potential for a little bit of conflict here. Females prefer to be at temperatures where they're able to exert more choice. Males prefer to be at temperatures where they're more likely to mate. All right, I'm going to complicate things one step further with a recent study. And that is because the previous experiment, we looked at female choice and male signal separately. Now we're going to put them together and measure them at the same time. So we assessed both the male signals and the female choice. In this way, we can actually assess what the females care about. What specific aspects of the male signal are they paying attention to? And we did a method to basically take the male signals, and we used what's called principal components. The details aren't super important, but you can just think of different combinations of aspects of the male signal got grouped into new variables. And I'm going to focus on this third one, which focuses on overall rates and durations of the display. So if we look at this variable, the rates and durations of the display, principal component number 3, you can see over the course of the two temperature treatments, it doesn't change. So the male rates and durations of the display don't have a significant difference. However, if we look at the female preference for this trait, if we look at hot temperature on top and room temperature on the bottom, something really interesting happens. So at lower rates and durations of the display, so with shorter displays, female probability of copulation is very high, whereas at longer durations of the display, their probability of copulation is lower. However, when we look at the cool temperature, there's no preference at all. So at hot temperatures, females want males to speed things up. At cold temperatures, they don't care how long it takes. So something very complicated is going on that we don't totally understand. And that's basically the conclusion I have for this. We don't totally understand this, but there's a lot of interesting interactions that happen. All right, and I'm running short on time, so I just want to very briefly give you a slight introduction as to how this could have bigger impacts on the entire species complex. So Hebrides clypeus lives here, kind of near the bottom of a mountain. But we know that all these different elevational sites are very different in terms of temperature from one another. And lo and behold, there are other species of spiders that live in all these elevational regions. And I did physiology studies on all of these species. And there are some interesting differences, although perhaps not what we might expect. So you can imagine if we see this interaction like we saw before, where females are less choosy at high temperatures and males are more likely to mate, is it possible that if climate change puts these species into contact with one another and they're warm, are females more likely to make poor decisions? Poor decisions as in, are they hybridizing with one another? We know that these species can readily hybridize with one another, and we're starting to see more and more hybrids. So this could really have some interesting impacts for the history of the species complex as a whole. So just to kind of summarize, males and females have different thermal preferences, and there's some really interesting complex interactions when it comes to temperature and mating behavior with potential for very broad scale implications. And that's all I've got today. A few acknowledgements, and I'll take any questions you guys might have.
Wonderful. Thank you very much, Erin. Any questions? If you,
It's actually Geronimoi. Which is
Geronimoi.
more fun to say, I think.
If
They're
you
also really
discover a
hard
new
to
one,
find. They're found in these super deep, like pine litter. So you have to like lay down in the pine litter and just like watch for these little guys hopping around. I think in 1 week of looking for them, I found 6 or something. They're so hard to find.
I have a question, just briefly. You said hybridizing between species, like, is like, does that have negative fitness consequences?
Potentially. So the lab I did my PhD work in, they're really going nuts with this. So they're attempting to do lots of different hybridization trials to see, like, females are kind of known to prefer signals from males that aren't in their species. And sometimes the hybrids are fine and we just find them. There's also the potential that hybrids could be sterile, or hybrids could be non-viable at all. And it seems like more closely related species hybridize more readily, as you might expect, but there's also some weird ones that happen. So I guess the answer to your question is we're not sure. There's certainly the potential for some pretty serious fitness impacts if individuals are wasting their time mating with individuals that aren't going to produce offspring.
Mm-hmm.
Oh, okay. I got a question here that I will read. Do hybridized spiders have a wider range of sexual interactions, or do the hybrid preferences pull in one direction? Boy, I would love to answer that question. And that's something that I haven't worked on. It was something I really wanted to do, and I just ran out of time, unfortunately. But that's something that's happening right now. And there's actually been a recent paper that came out that kind of mapped all the different hybrids that are found in these species. And it's a giant phylogeny, and the lines that show the hybrids are just crazy. They hybridize all over the phylogeny. And I don't really know about like the sexual interactions of those specific hybrids, but that'd be super interesting to look at. They are very tiny.
I'm still trying to puzzle through the mate choice results. How do you measure, like, so you can measure the kind of their preference on that thermal gradient in, I guess you measure that independent of any social context.
Right, we isolate them. So there were little lanes on there, and we just
Okay.
put one. So something interesting that I didn't show is that we also, I did a field experiment where I did focal follow observations of 100 spiders for 15 minutes each. And we measured their temperature in the field. And that difference between males and females completely disappears. So I suspect it could be because in the field, males are typically chasing females down. So females are the ones that actually get to choose the temperature, even though males have kind of an innate preference for higher temperatures. So it's kind of an interesting exercise in what do they actually want versus what are they able to do in the field.
Do you have a way to know what is the best kind of choice environment for females, or is the assumption just that if she's doing more rejections, that's she's exerting her preferences more?
That is a great question. And this is a really challenging system to do that and to do choice trials in general, because the females mate once, so you can only run each female one time. So you kind of have to do preference on a population level rather than on an individual level. I see studies with crickets and frogs, and I'm just so jealous because you can ask females really directly. They also don't tell you what males they like unless they're physically mating with the male. That said, there is some known about different species of spider, and females do have kind of environmental preferences. So my PhD advisor worked with peacock spiders, and they were trying to get these spiders to mate, and they couldn't. And they finally realized that the female required a small stick in the middle of the courtship arena. And without that small stick, nothing would ever happen. So there's definitely some weird stuff going on. Habernatus are pretty well established as you put them in an arena and they'll more or less do their thing. But yeah, that's certainly interesting to look at.
Thanks. All right, any other questions before we sign off? Okay, I want to thank our 4 speakers again. I really appreciate it. Yeah, thank you for these presentations. Very interesting. Really neat to see.
Thanks for the invite. This was really fun.
Okay, take care, everyone. Bye.